Today we will discuss or examine about the pain sensation or nociception. Here is the summary of the pain sensation in this diagram, which I would be summarizing during my presentation. I am dealing this particular chapter in 3 different lectures. In today's lecture, I would be focusing on the definition, the receptors, the pathway, and the representation. In the next lecture, I will look into the processing component of the pain at various levels. In another lecture, I will talk about the pain management strategies. Most of the patients, they come with pain as one of the primary symptoms. For that reason, this becomes a very important aspect as far as the patient concerned, because if there is no pain, he may not come to you at all. What is the definition of a pain? Sir Sherrington defined it as the psychical adjunct of an imperative protective reflex. Later on, the International Association for the Study of Pain has taken up the task, and they defined it an unpleasant sensory and emotional experience associated with actual or potential tissue damage or injury. However, somewhere after 10 years or after 20 years, the International Society Study of Pain, International Association for the Study of Pain, they sat together and they redefined this particular definition. Now, currently, this definition is the pain is a distressing sensory and emotional experience associated with the actual or potential tissue damage or injury. If you are coming, pain is a distressing. So the word distressing, they have replaced that unpleasant component. It's the distressing component they added. Of course, there were a lot of discussions on this issue, and I'm not going into the detail of that. Now, what are the receptors for the pain? For the pain, the receptors are the free nerve endings. There are no defined encapsulated nerve endings. These are free nerve endings, and these free nerve endings located in all parts or all segments of the tissue, including the viscera. And these free nerve endings are carried by one fast fibers, myelinated fibers, thinly myelinated fibers, A delta fiber, and this A delta fiber is about 5 microns in diameter, and the conduction velocity ranging around 10 meters per second, 2 to 10 meters per second. And the unmyelinated C fibers, these C fibers are having a conduction velocity less than around 1 meter per second. Their diameter is around 1 to 2 microns. Now, thus, because of these two components, thus pain sensation can be divided as a fast and a slow pain. Fast is carried by the myelinated fibers, that is thinly myelinated fibers. The slow is carried by the unmyelinated fibers. Now, what are the stimuli or a stimulus for the nociception or pain? Nociception and pain are used synonymous. Nociception is a sensation of unpleasantness or noxious, injurious sensation. That is a nociception. So nociception or pain, what are the stimuli? There is no single stimuli for the pain sensation. So say, for example, for a touch, it's a mechanosensitive. For vision, it is the light. For sound, it is mechanical. For temperature, it is thermal. But in case of pain, it is not single, it is multi. That is why it is called a polymodal sensation. Polymodal sensations, that is, it is being carried by the different stimulus or stimuli. One, the mechanical. The mechanical is suppose if cutting, crushing, pricking, tearing, stretching. What is happening? You are just separating the tissue, or the tearing the, or rupturing the nerve fibers that would produce the pain. This is mechanical. Then we have the chemical stimuli. These chemical stimuli are the various the products of the cell injury products. Because of the injury here, several cell injury products. What are these cell injury products? The first one is the potassium, because the inside, if the cell is injured, whatever is present within the cell is coming out. That is potassium, hydrogen ion. Then we have number of chemicals within the cells like histamine, prostaglandin, serotonin, bradykinin, and the ATP molecules, the adenosines, the cytokines. Then we have some peptides like calcitonin gene regulating peptide and substance P, catecholinins, and other inflammatory mediators. They form the chemicals, the number of chemicals. They will be, and they will be stimulating the pain receptors. Then we have a thermal sensation. The thermal sensation, because yesterday we just looked into the thermal sensations. The thermal sensations on either side, whether it is a cold, extreme cold or extreme heat, produces the noxious or a painful stimuli. So that means even a temperature when it goes above 45 degrees Celsius, or a temperature when it falls, maybe as it falls below 15 degrees Celsius, or maybe 10 degree or below, it starts feeling the pain. And the high intensity stimulus to all sensory receptors. Any receptor, if there is a too intense stimulation, then what happens? So it would produce the pain. Say, for example, if a sound, very 120 decibel sound or 140 decibel sound, that produces pain. Very sharp and focused high intensity light, that produces pain. Or any pressure effect on the pressure receptors, that produces pain, or a touch, or any modality that would produce the pain. Primarily there are three major components of major mechanical major stimuli for the pain: the mechanical, chemical, and thermal. And non-specific is high intensity stimulation. Now, so regarding the thermal nociceptors, because yesterday I looked into this thing, these polymodal thermal nociceptors, these are here, and especially heat sensation, the TRP V1. Yesterday I was talking about the TRP V1 and V2. They start activating somewhere around or after 42 degrees Celsius, and they become 45, and after 48 degrees it becomes painful. So these are the TRP V1 and the TRP channels. These are TRP V1 channels. These are heat sensitive. And thermal cold sensation. Cold sensation is coming from this TRP menthol, TRP M8, or enchirine, TRP enchirine. These are the TRP M8, TRP enchirine channels are not shown here. Somewhere here we have that. And same thing when it comes to polymodal, we have a peptidergic and a non-peptidergic components. The peptidergic components are stimulated by the substance P, CGRP, neurokinins, and all other peptides. They are stimulating these peptidergic free nerve endings. The non-peptidergic chemicals include the potassium, hydrogen ion, prostaglandins, lipids, ATP, etc. So all these things stimulate these things. In addition, this mechanical transduction that also produces the pain. So as far as coming back to the thermal sensations, we have the thermal heat receptor and a cold receptor. And yesterday I just discussed this chart with you. And in this chart, now today what I have made this chart here. Here I have made, this is heat. As the temperature increases above 45, above 45, so these are activated, these nerves are activated. The heat, the fibers producing the heat pain are activated. Maybe yet or above 48 or 49, you just see that this is the range, this is the change already, this intensity of the pain. This is, I'm indicating this color code as the intensity of the pain. As it's starting from here, it is you don't have the pain here. Slowly changing, the warmth is felt, and the yellow is the changeover from the slightly you will have the warning signal. That means you may have a little pain or burning. Then as it increases further, this burning becomes red hot. So you can just see that red hot. So this is the increase in case of the increased temperature. Similarly here, what happens is somewhere around 10 degrees Celsius, somewhere around 10 degrees centigrade, it is the yellow is, that is the point where you start feeling pain. And as it decreases further, you see that redness is increasing. So this is the type of effect on the nociceptors, how it is, this is a painful area, this red is the painful area, this is the painful area. Now, what are the chemical mediators? Here is a scalpel, you cut it, or it is just a cutting. What happens when you cut? It releases the intracellular products, that is the potassium first. And along with the potassium, hydrogen ion is there intracellularly, that would stimulate the free nerve endings and produce pain. And also releases a number of chemicals. These number of chemicals are prostaglandin, serotonin, bradykinin, and histamine. And this histamine also comes from the as an inflammatory response, because you are producing an injury and that produces an inflammation. And the inflammatory response, the mast cells, the tissue mast cells, they release histamine. So that means that produces the pain. And also the mast cells also release a number of chemicals like interleukins or cytokines or other chemicals. They even including the prostaglandin and bradykinin. These are coming through the mast cells. So this is what this is here. And these mast cells in turn are activated by the neurotransmitters like substance P or CGRP. CGRP is a calcitonin gene regulating peptide. They will activate the mast cell to release histamine. At the same time, you have these transmitters released here, the CGRP, substance P. They will activate the blood vessels. They constrict. And they will produce. So that means all these chemicals activate these nerve fiber, and they will send the information to the spinal gray matter or the spinal neurons. Now, here is the receptors for the pharmacological receptors for the nociceptor signaling. So these are all pharmacological receptors on the cell membrane. On the cell membrane, if you are looking at that here, these type of receptors or ion channels. So on these cell membranes, what are the various pharmacological receptors? So these are various chemicals when an injury takes place. This is a macrophage, and this is a tissue damage, and tissue damage releases H ion and potassium, and also ATP, and then adenosines. So they will activate. They will activate here. I will just come back one by one. The H ions, this activates these receptors. Let me tell about the receptors first. So we have a number of chemicals which are released because of the inflammation, and these chemicals are known as the chemicals of inflammation or inflammatory soup. And these chemicals act on the self-surface receptors. The self-surface receptors are a G protein coupled receptor. Some are G protein coupled. Say, for example, bradykinin, prostaglandins, histamines, nerve growth factors. They act through these GPCR. Or there are other channels, that is the transient receptor potential channels, that are for the vanilloid TRPV, various components V1, V2, V3, V4, V5, or TRP menthol, TRPM. So these are TRP channels. They will activate on the TRP channels. They will be producing the pain. Or you have this cold, that's the enchirine, TRPA. So these are transient receptor potential. These are transient receptor potentials. Then we have acid sensitive ion channels, ASIC. This is acid sensitive ion channel. This will detect the acid or the pH change. And we have a purine receptors. So purine is the ATP or adenosine related. So P2X. The P2X receptors, these P2X receptors are activated and they are producing the, they are activating here. Then we have the potassium channels. These are called a two-pore potassium channels. So there are two pores here, one pore here, one pore here, two-pore potassium channels known as K2P. And we have tyrosine kinase receptors. These are receptors for tyrosine kinase. So that means we have nearly 5, maybe more number of receptors, pharmacological receptors or ligand-gated receptors, which will detect the inflammatory chemicals. These inflammatory chemicals like the interleukins, nerve growth factor, histamine, bradykinin, prostaglandin, adenosine, and ATP, and these are being produced by the macrophage, mast cells, tissue damage products, from the platelets, from the immune cells. Okay, these are the various signaling, and once they enter, once they trigger these cells, they produce the intracellular signaling, and by producing the intracellular signaling, they will produce the depolarization and produce an activity or an action potential. Now, what are the various, here I am just summarizing the nociceptors, their peripheral axons, and the information they carry. One, the receptor type. Here the free nerve endings, receptor type, free nerve endings. The fiber type is carried by the myelinated, thinly myelinated A delta fibers. They belong to group 3. And their conduction velocity is in the range of 5 to 30 meters per second. I will keep 10 meters as on an average, because that is the range it is coming around. And the axon diameter of these free nerve endings or the receptors is between 1 to 6 micrometer. And what they do, they detect the sharp pain. They detect the pain arising from the cold. Then we have free nerve endings. They are supplied by the type 4 or C fibers, unmyelinated. And their conduction velocity is slow. It is around 1 meter per second on an average, and their diameter is around 1 micrometer or less than 1.5 micrometer. These carry dull, aching pain and touch, pressure, or warm. So these are the things which these carry free nerve endings. So these sharp pain is activated by mostly by the sharp pain is mostly activated by the mechanical things, and the slow pain is by the chemicals. Now, the first order neurons, say for example, A delta fibers and C fibers, they reach here in the spinal cord, laminae 1 and 2. This red one, that is the C fiber, is laminae 1 and 2. And the A delta fiber is laminae 1, 2, and 5. So you can just see that it's a substantia gelatinosa cells making contact with a wide dynamic range neuron. This wide dynamic range neuron is a projecting neuron. Wide dynamic range neuron. It's a projecting neuron which ascend as a lateral spinothalamic tract. Now, what are the differences between the fast and the slow pain? Just I'm trying to summarize whatever I have made up till now. The fast and the slow pain. Now, fast pain, it is known as a epicritic pain. The slow pain is a protopathic. Fast pain is sharp, pricking, acute, or electric pain. Slow pain is burning, aching, throbbing, nauseous, that is a visceral sensation, and chronic pain. The receptors here are free nerve endings. Here also free nerve endings for the slow pain. The fast pain is carried by the A delta fiber with a conduction velocity 5 to 30 meters per second. The slow pain is carried by C fibers, unmyelinated, conduction velocity 0.5 to 2 meters per second. The fast pain is activated by the mechanical and the thermal stimuli, and slow pain is usually a chemical stimuli. It is relayed into, the fastest pain is relayed into 1, 2, and 5 laminae, and it is relayed in laminae 1 and 2. This is known as a neospinothalamic projection. The fast pain is a neospinothalamic because it is going into the cortex, the cortical area S1, and trying to discriminate the sensation. Whereas, and it is for the precise localization and providing attributes and define particular pain originating. Here, in case of it is a paleospinothalamic, it is diffuse, non-specific, non-adapting. It does not adapt. And if the pain adapts, it is not. Then it is relayed. It is not relayed into the somatosensory area 1, the post-central gyrus. It is relayed through the amygdala to the hypothalamus, the insular cortex, and the cingulate cortex. This is for the behavioral and autonomic responses. Now, as we have all experienced, suppose if there is a cut to you, whatever action you are doing, you have a cut. What happens? Suddenly you will have a pain, and this pain is sharp, and you will withdraw it from the hand. And after withdrawing it from the hand, most of the persons are trying to make this thing, trying to avoid or trying to move the limb, so that will trying to take away from the site of injury. Then after some times you will feel there is a dull ache. If you have not experienced, whenever you may be knowing it, but you are not aware. So dull ache comes all through after some time. And this dull ache is a slow pain. So that means whenever there is a mechanical cut, there will be immediately a epicritic touch, then followed by the chemicals released, that's a protopathic touch or protopathic pain, epicritic pain and a protopathic pain. So now the pain fibers relay in the spinal cord, as I have already mentioned. These are nociceptors, A delta fibers 1, 2, and 5, C fibers 1 and 2. And what happens? They come to the spinal cord. They are not relayed immediately in the same segment. Some of them ascend up 1 or 2 segments. Some of them descend up 1 or 2 segments, and they form a tract known as a Lissur's tract. Yesterday I was trying to talk about that Lissur's tract. This Lissur's tract is a pathway formed from the proximal end of the A delta and C fibers. These are A delta fiber and the C fiber, and they will go 2 segments up and 2 segments down, and then terminate in the designated laminae 1, 2, 5 laminae of the spinal cord. That's what the relay, and I mentioned about the Lissur's tract again. Now coming back, after the relay, say for example, if I am coming here, this is the relay here. this is the wide dynamic range neuron, and this is where from 5, this is being relayed, or it will cross to the opposite side. This is anterior to the central canal, and ascend as a lateral spinothalamic tract. So that is for all the sensations which are epicritic pain sensations. But the pain is carried by at least 3 major ascending pathways: spinothalamic, spinoreticular, spinomesencephalic. Whether they are carried in a whole, something like this, or because of the collaterals, there will be more. So not all the pain fibers, the most of them, they will be going into the lateral spinothalamic tract. Nearly 10% or so, they will be ascending as a spinoreticular and a spinomesencephalic. These lateral spinothalamic tract, again, you have the paleo and the neo sensation. Neo sensations are ascending in the lateral spinothalamic tract. The paleo sensations are ascending in the spinoreticular and the spinomesencephalic part. So coming back here, the pain fibers after crossing, they will ascend as a lateral spinothalamic tract. They will ascend, they come to the thalamus. In the thalamus, the ventroposterolateral nucleus of thalamus, and from here it will reach to the somatosensory postcentral gyrus somatosensory area 1. From the somatosensory area, it will try to localize, it will try to give the attributes of the particular, whether it is coming from a cut or a heat or from any other aspect, the bite from an insect or whatever. So now that is being discriminated. This is for epicritic, for identifying where it is coming, and it is usually a fast pain. Okay, that's one part. And the second part, the spinoreticular, what is happening here? It is coming here, it is ascending up, and it gives a relay into the reticular formation, medullary reticular formation, the pontine reticular formation, and the mesenchymal reticular formation, the medullary, pontine, and midbrain. And this the medullary reticular formation, they are necessary for the responses, especially the respiration and the blood pressure, their regulation, because if there is a pain, it is associated with the alteration in the blood pressure and respiration. So that means that is one thing which modulates. This is one aspect. The second aspect, this reticular formation sends impulses to the thalamus, reticulothalamic tract, and this reticulothalamic tract, that would be for arousal response or awake response. That would go to the entire layers, all the cortical layers for arousing. So that is the thalamocortical projections all through. So that is from the reticular formation. This pontine, that is upper pontine nucleus, there is what is called a parabrachial nucleus. And from the parabrachial nucleus, it goes to the different nuclei of the dorsomedian nucleus and the ventroposterior nucleus, ventromedian posterior nucleus. So these we were discussing about, and from here either they are connected to insular cortex or connected to the cingulate cortex. This is a cingulate cortex here. This is going here, and here then we have a third pathway, the spinomesenchymal, especially this pathway to the periaqueductal gray. So that means they will give the neurons present around the periaqueductal circle or the circumference. This is the aqueduct, is the ventricular, these things around that, it gives the there are neurons, scattered neurons, it gives there. And these neurons, actually, they have encephalinergic neurons. These try to modulate the whether this should be allowed or this should not be allowed. Then they would also project onto the thalamus and hypothalamus to look after. So that means this is a spinomesencephalic. The spinomesencephalic, these things goes to the periaqueductal, and that will try to modulate the pain either at this level or sending it descending or ascending influences. And similarly, the reticular things also modulate the spinal activity. So it is not simple. If you are thinking about a medial lemniscal pathway, the medial lemniscal pathway is simple, is going up in the to the medulla, and where the nucleus gracilis and nucleus cuneatus they relay, and after that they cross over and ascend as a medial lemniscus, reach the VPL of thalamus and project onto the somatosensory area and perform that function. So that is precise one. Here it is a multi-projections, and these two form the paleo spinothalamic tract or paleo. They will take care of the paleo sensations. This is a neo sensation. Now going further about the epicritic pain. So now for the epicritic pain, it is the receptor, and this receptor is carried by the A delta fibers. A delta fibers reach the spinal dorsal horn neurons 1, 2, and 5, and they relay here. From here, they ascend, cross, and ascend up as a lateral spinothalamic tract. Reach the ventroposterior lateral nucleus of thalamus and reach to the somatosensory cortex. This is somatosensory cortex. So this is I have made a destination from the receptor, free nerve endings. Usually it is mechanical or thermal, cold. Then layer 1 to 5 here in the spinal cord. Then cross number 3 here in thalamus, the VPL, and then cortex S1. That is the thing. The remaining part, the collaterals to parabrachial nucleus, I was trying to impress you upon, engage a cingulate and insular cortexes and amygdala. So that we will see in the next slide or subsequent slides. But this projection, whatever is going from the thalamocortical projections, they will just be shared, or they will be just distributed following the sensory homunculus, which we have seen earlier. So that means we have a greater representation, the parts of importance, say for example, the fingers, the hand, the face, the lips, the vocal cord, not the vocal cord, the pharynx, the tongue, and very little importance to the intra-abdominal structures and genitals and other segments. So that is the typical homunculus for any, even for this lateral spinothalamic projections, similar to that which has been sent to the going up with the medial lemniscal fine touch. Okay, but the representation is not as dense as that of the lemniscal sensations. Now, the second part, which we were trying to worry about, so the same thing I have brought here. This is a paleo spino or paleo sensation, or the pathway for paleo sensation, and this paleo sensation is for arousal and behavioral changes. This is a protopathic pain. Primary nociceptors or efferents relay in the spinal gray matter. These are nociceptors, mostly C fibers and free nerve endings. That epicritic is for mostly A delta fibers. Here it is C fibers. They will come relay here in 1 and 2, area 1 and 2, and then cross and ascend as a spinothalamic tract. Okay, it is going up to the thalamic tract. Okay, that is broadly, this is not as much, but the major contribution, it gives collaterals to the rostroventral medulla, then mesenchymal, that is around the periaqueductal gray. Then we have the upper pontine, the parabrachial nucleus. Parabrachial nucleus. And all these things ascend up. Now, the information given to the parabrachial nucleus assumes a great importance, because from here it is connected to amygdala, it is connected to hypothalamus. So they have shown only amygdala here. It is also connected to hypothalamus. Parabrachial and hypothalamic connections are there. And amygdala and hypothalamus are interconnected. So now parabrachial nucleus to amygdala, parabrachial nucleus to hypothalamus. Hypothalamus brings in the autonomic responses, and the amygdala will produce the emotional responses. In addition, the parabrachial nucleus also gives a descending these things to the sympathetic and the parasympathetic, that is autonomic centers. So from amygdala, the fibers reach the insular cortex and cingulate cortex. That's what I have written here. The primary nociceptor afferents relay spinal gray matter, cross, and ascend as a spino-reticular tract, relay in a reticular system. This is a reticular system there. This is entire thing is a reticular system. And fibers reach the intralaminar or internal medullary nucleus and the reticular nucleus of thalamus, and they will project onto the entire cortical areas for arousal response. The relay in the parabrachial nucleus reach amygdala, cingulate cortex, and insular cortices. And that would produce the behavioral response, what to do, how to react, and how much to be. That is another aspect associated with the pain. Pain, the behavioral or emotional response is associated with the pain. The release in a rostral ventral medulla and this part and the periaqueductal gray provide feedback signals for the pain control. So this one here only it may control or send projection to the spinal cord to control the pain. So the same thing here, reticular system that would also project onto the spinal thalamus as a bulbospinal tract that is coming from the raphe nucleus, the bulbospinal tract, or a periaqueductal gray, which is activating the bulbospinal tract, and that is for the pain control mechanisms. Now, what are the various neurotransmitters involved in the pain pathway? Now, if I were to talk here, the neurotransmitter here, the nociceptor, this is one synapse. This neurotransmitter here is a glutamate and the substance P. These are the major neurotransmitters. So I have written here glutamate is an excitatory neurotransmitter. Then a substance P is an excitatory neurotransmitter. Then we have from the dorsal root ganglion, we have another interneuron. This would release the inhibitory thing, that is enkephalines or endorphins. These are known as the endogenous morphine or endogenous opiates. And these neurons, they receive input from descending things, serotonergic inputs, the 5-HT, norepinephrine inputs. You have a norepinephrine inputs. So then here in the segment here, there are inhibitory components. These inhibitory components having a GABA, that's inhibitory transmitters. Then they will also activate the histaminergic neurons and glycinergic inhibitory neurons in the spinal cord. These are the various neurotransmitters involved in the pain pathway. So now these are the other neurotransmitters like substance P, the neurokinin, adenosine, that is ATP. They will either produce the inhibitory or excitatory tachykinins, mostly excitatory, and PGE2 excitatory or inhibitory, nitrogic, that is a nitric oxide, that is either excitatory or inhibitory, CGRP, the calcitonin gene regulating peptide, that is excitatory, and cannabinoids. Cannabinoids are TRP, capsaicin, the things which respond to the capsaicin, these are cannabinoids. So these are some of the neurotransmitters involved in the pain pathway. Now, what is visceral pain? Visceral pain, as we have already seen, this is not the same as the cutaneous pain or somatic pain. Usually, the representation of the abdominal or pelvic or thoracic viscera is poor in terms of the localization. Now, this representation, say for example, the heart. So this is the part where the heart, whenever there is a problem in the heart, you can just see that this particular component. Whenever heart is affected, depending upon the area it is affected, you have the epigastric pain, or on the shoulder or the neck, then it is radiating down. The arm here, you just see here, radiating down here. This is the pain related with the heart. And if you are looking at this part, so this is esophagus. Esophagus is here, and this esophagus is having epigastric pain, and also it will have a similar, almost up to this point, it has a similarity with the heart. So that means anything happening to esophagus or esophagitis or GERD, you may end up with the symptoms similar to the heart pain. So this is esophagus. This is stomach. You have the epigastric pain. Just see the epigastric pain. The liver here on the right side, right hypochondrial area. Then the pylorus here in the upper or in the middle compartment. So then the umbilicus in the center here, and the kidney may be on right kidney on this side, the left kidney on this side, and this is a urinary bladder. This is urinary bladder, and even appendix and small intestine. And we have the ureters representing down here, and in the perineal area. So that means these visceral pain or referred pain or refer to the dermatome the organs are being originating. The visceral sensations belong to that. And the various different organs as if something is happening outside. Why? What is the phenomena? What is the physiological basis of the referred pain? So now here, what happens? The visceral pain usually results from a stretch or inflammation. Say, for example, if the intestine is stretched, so that produces or inflammation of the intestine that produces this is the thing, viscous. That is the viscous. And it goes here, the information through the sympathetics, the reach to the ascending fibers, the spinothalamic tract or the projecting neuron, they activate, they converge on this. This is the dermatome pattern which is coming up. So that means it sets the same neuron from the same dermatome. That is why you feel, or the person feels a pain originating from this side. Because the neuron going here, it will go to the brain, and it will be in the somatosensory area. It has been identified that this is the segment where it is originating. That is the basis, and this is a convergence theory. So that means the visceral neurons, the sympathetics, they are coming here, the visceral afferents, they are coming here, projecting or converge on the same neurons originating from the dermatome. So that is the convergence theory. Some of them, they believe that they have a separate line, but the separate line While ascending up, they will also produce or excite the dermatomal neurons. This is a facilitation theory. So that means this will facilitate while going ascending up, it will facilitate, so that would produce the pain. Okay, so this is the basis for physiological basis for referred pain. Usually it is the convergence theory. Nowadays it is mostly convergence theory, and it is also facilitation, as it is you see here, the visceral pain is going up here, and it is sending in impulses. Simultaneously, it is also facilitating the dermatomal, and you will feel the pain in the particular area of dermatome originating from. What are the different types of pain? We have three different types of pain: nociceptive pain, neuropathic pain, inflammatory pain. The nociceptive pain is resulting from the activation of the A delta and C fibers. Both of these nociceptors are specialized, originating from the free nerve endings located in various tissues, the skin, muscle, joint capsule, bone, or any other internal organ. They functionally detect potentially damaging chemical, mechanical, and thermal stimuli, which is harmful, and carried by the pathways mentioned for both epicritic, that is discriminative, and protopathic modalities. Epicritic pain, that is a discriminative, identify the location and the attributes, and the protopathic is about the emotional components of the pain. That's about nociceptive pain. Then neuropathic pain. Neuropathic pain is commonly seen in nerve injury or impairment, and it is often associated with allodynia. What is allodynia? Allodynia is that the particular sensation, a touch sensation. Now the receptors or the area carrying the touch sensation has become painful. If I were to put that, it is a central pain sensitization. It is happening at the central level as a result of a repetitive, non-painful stimulation of the receptors. Repetitive and non-painful stimulation of the receptors. It triggers a pain response from the stimulus that is non-painful. That means it may be touch, pressure, or any other modality. And it is due to the sensitization of the nerve fibers ascending up. It's not happening at the peripheral level. It's happening at the relay level or the central level. This condition can be described as a pathologic pain because a neuropathic pain is not a protective. It's not a protective pain. Pain could be continuous sensation, or it may come as episodes. Because of the injury, sometimes it may disappear and again come in another episode. The major causes of this pain are primarily due to the inflammation or metabolic inflammation of the nerve fibers, or the metabolic diseases which affect the nerve fibers, like diabetes, trauma, toxins, tumors, or even the viral infections like herpes zoster and other primary neurological disease. This is also known as a casualgia. Casualgia, that means that produces the pain, or a thalamic syndrome, or trigeminal neuralgia. So these are the examples of the neuropathic pain. Inflammatory pain comes from the inflammation, that is the site of injury. It may be acute or chronic. Acute inflammatory pain is intense and lasts for a short period and is initiated by noxious stimuli involving A delta fibers. The chronic pain is vague, dull aching, and lasts longer. These are non-adaptive, initiated by chemicals or thermal modalities involving C fibers, that is slow. The inflammatory pain increases afferent input to the spinal neurons and produces sensitization. That is a central sensitization. So that means inflammatory mediators from the site activate the nociceptors within the inflamed area. So that means the inflammatory mediators, say for example, we have a um, serotonin, we have a histamine, we have a nitric oxide or any other potassium or hydrogen ion, all of them are adding up to the excitation of the nociceptor. So that is what these nociceptors are activating. That means they are complementary to each other, synergistic. The chemicals include cell injury products, as I have already mentioned, such as potassium, hydrogen, ATP, 5-HT, kinins, histamine, nerve growth factor, adenosine, ATP, and adenosine triphosphate, prostaglandin, glutamate, leukotrienes, nitric oxide, and norepinephrine, and others. Produces what is called, because sensitization of the receptors, the peripheral receptors, this is known as a primary hyperalgesia. It is because of the excessive sensitivity of the pain receptors by the inflammatory mediators or by the increased temperatures. Because inflammation is associated with the increased temperature. Now, allodynia, I mentioned, it's a pain evoked by a stimulus that usually is not painful. That is called allodynia. While hyperalgesia is an increased response to the normal threshold. This is happening at a receptor level. This is happening at a central level. The nervous pathway for pain sometimes become excessively excitable, and this gives rise to hyperalgesia, which means hypersensitivity to pain. So that means this is a secondary hyperalgesia. The primary is the receptors, the pain pathway is being sensitized, that is secondary. So now here I am trying to differentiate primary hyperalgesia or excessive sensitivity of the pain receptors. This is a pain receptor. That means that the sensitivity of the receptor is increased. There are many inflammatory mediators. These receptors are activated. Pain is going up. That happens in case of inflammation. The secondary hyperalgesia, facilitation of the sensory transmission. Say, for example, here, here, or here. So at 2, 3, and 4, the transmission, because of the increased excitability of the neurons here and here and here. So that is secondary hyperalgesia. The summary, it's a pain is an unpleasant sensory and emotional experience associated with the actual or potential tissue damage. The receptors are free nerve endings. These are nociceptors, free nerve endings carried by C fibers and A delta fibers. Now, once they are carried here, some of these fibers will activate the muscle reflexes, that is withdrawal reflexes, because you have to withdraw from the site. So this is the pathway for withdrawal reflex. These are muscles. This is a withdrawal reflex. Then others, they are relayed into lamina 1, 2, and 5 in the spinal cord. Lamina, you can see the lamina here, 1, 2, and 5 in the spinal cord, and cross to the opposite side, in front of the central canal, and ascend as a lateral spinothalamic tract. While ascending lateral spinothalamic tract, it gives collaterals to the rostroventral medullary reticular nucleus, the pontine medullary reticular nucleus, that is the parabrachial nucleus, and the mesenchymal area, that is a periequidectal gray. Okay, so the epicretic touch, which is reaching through to the thalamus at the VPL, that is ventroposterolateral nucleus, reaches the sensory cortex S1, and that is for localization and attributes of the modality of pain. The protopathic component that comes from the parabrachial nucleus, and from this parabrachial nucleus that would go to amygdala, and then from amygdala to the insular cortex and the cingulate gyrus. The anterior cingulate gyrus, they will be for behavior and other reactions. So these insular cortex and anterior cingulate cortex, they are connected to the prefrontal cortex, and also to the hypothalamus. In addition, the parabrachial nucleus has a connection with the hypothalamus. Reticular projections, that means these are the reticular projections, they reach the intermediolateral nucleus of the thalamus, and they are for arousal response. Reticular and mesenchymal, this is a reticular and mesenchymal projections. They produce a descending inputs and the local inputs. They try to modulate pain at the site and the descending projections. This is about the summary of the same pain receptors, the pathway and representation in various areas. And these are the reference books. Some of the figures are taken from Candle, and I have mentioned whatever I have taken. These are some of the, I have taken borrowed it from the latest literature available. These are some of the articles I have referred, and this is one of those major this thing. Okay, so now I have a lot of assignments here. Maybe I just read it for you. Maybe you may have to do this thing, because this is an important topic. It comes in the NTR University very often. So I read those topics: define pain, describe receptors, pathways, representation of pain sensations, write the differences between fast and slow pain. Please prepare. Then I have written here the short notes on the fast pain, slow pain, the nociceptors, the inflammatory soup, the polymodal receptors, draw a diagram of spinothalamic tract, spinoreticular pathway in pain, spinomesenchymal pathway in pain, pathway of protopathic pain, allodynia, neurotransmitters involved in pain transmission, hyperalgesia, primary hyperalgesia, secondary hyperalgesia, neuropathic pain, physiological basis of visceral pain, inflammatory pain, nociceptive pain, differentiate acute pain from chronic pain, differentiate primary and secondary hyperalgesia, and differentiate fast pain and slow pain. I think I have, if you do this thing and you are thorough with this topic, okay. Wednesday or next class, I will discuss about the pain processing at various levels. Okay, thank you.